All right, perfect. Thank you for letting me present here, Dennis. So I'm a third-year PhD student in Jakob Jespersen's group here at the MGH on the Broad, and I will tell you how we can use protein structures in understanding missense variation. So first of all, I will tackle two questions. What's a protein structure? So a protein structure is somehow experimentally resolved three-dimensional static model of a protein. And so how is a protein structure made? So the structures that we can work with, they are obtained by one of different methods. So the most common is X-ray structure. So a crystal is made and the structure is solved. Nutrient diffraction is also coming up, and we have other methods like NMR or cryo-EM. And then there are some predicted methods. So it's possible to make homology models if you have an existing protein structure and you have another protein with a similar sequence. You can use those bits from the existing one and patch together a new model. And recently it's also been structures have also been predicted with coevolution. So if two amino acids have co-evolved, there would be a distance constraint and they would be solved in a similar manner to NMR. So there is this competition called CASP, Community-Wide Experiments in Critical Assessment of Techniques for Protein Structure Prediction. And it's really helping advance the protein structure prediction. And you can go there and see a lot of groups trying to be the best at predicting. So to the second question, what is a missense variation? So in the genome, when you have a single nucleotide variation resulting in the translation of a different amino acid. So the coding for one amino acid changes and you'll get another amino acid. So why are we interested in doing this? So understanding human genetic variants better can help us understand diseases better. So two examples are we can use the proteins to find drug targets for those proteins, and knowing which variants are damaging can serve as a reference for early DNA screening, for example. So in terms of interpreting mutations, I will just highlight a few methods, but there are a lot of different ways. So there is for example, this probabilistic approach that Caitlin and Mohab has been published, and there is this more brute force approach by Daniel MacArthur, and here I've shown the GitHub from Konrad Kaszewski's implementation of the Lofty plugin, which was also used in the Exact project. So I also wanted to highlight this journal club because there are a lot of good implementations among you guys here. How to understand the genetic variation. So what's up with missense mutations? So they can be a little bit difficult to put in a box because they can be anywhere from damaging to deleterious. And we even have this abbreviation VUS, variant of uncertain significance. And since missense mutations often end up in this category, what we thought was maybe we can use the existing protein structures to help assess this. So I will now go through an example of why this can be relevant. So here is an example of a biological function that is disrupted by mutation in a protein-protein interaction interface. So this example is from a paper from 2013 about a congenital gonadotropic hypogonadism, where two proteins are needed to have an interaction. And you see on the right, that's FGFR1, a receptor, and on the left, that's a ligand. And patients with a mutant in this amino acid, the arginine 177, they instead have histidine there. They seem to have this phenotype. So in the paper, they show how luciferase assay shows the activity based on the protein variant. So if you follow the dark blue curve here, you can see that when FGF17 is added, the activity will go up to 100%. However, if you look at the green line here and the red line here, you'll see that the activity is much lower. And the green line is this particular amino acid that mutates to histidine, and the activity doesn't reach 100% at all. The purple line here, you see, is just next to the wild type activity, and it's a different mutation that is not thought to be involved in the disease. And I just quickly want to show a cutout from the exact browser, and it turns out that the purple one, the one that doesn't make any difference, it is observed in a somewhat healthy individual, whereas the two ones that disrupt the activity, they have not been observed. So this is an example of some rationale behind looking into the protein structures. And there are different features we can understand from the protein structures, and interaction to another protein could be one of these features. So some working tools are already in place to assess variants with protein structures in a way or another. So two popular ways are polyphen2 and mutation assessor, both of which use a three-dimensional structure. Mutation assessor uses protein structure in a higher degree than polyphen2, but there are many more tools that use protein structures in assessing variation in some way. So when we use proteins, we are somehow limited of the data that's available, because we have information about 20,000 protein coding genes roughly, but how many of them do we actually know the structure of? So depending on how much coverage you want to have on these protein structures, you will have between 3,500 and maybe 6,000 different genes covered. These protein structures, they are biased. So in order for them to have an existing protein structure, they need to be interesting enough for people to put them in their grants and have a PhD student work on them, or crystallize a protein and solve this. So usually they're biased towards the well-studied proteins, but they're also biased towards proteins that we can actually solve with current techniques. So I think it's just 10 years ago, we didn't have that many transmembrane proteins, but they're getting more common to find now in the protein data bank. So in the data that I'm working with, we have 6,440 different consensus CDS ID. So this is just an ID describing one single isoform of human protein. We have 1.2 million different codons in the human genome with protein structure, and these are from resolved protein structures, and just over 90,000 of those are in interfaces. So this is all a lot of numbers. And I'm not sure how familiar everybody in the journal club is with the quality. So I just want to go through two important points when looking at quality. So one is the resolution, which you might have seen, and you may be very well aware that it's how much can we see detail. So I thought Google Map would be an interesting analogy. You can see much more detail in a high resolution, so that's lower resolution. So 1 angstrom is a really, really good one. 2 is also pretty good. But we don't want to work with all structures. We want to discard the worst ones. So if you didn't see it, the map is zooming in on where we are hosting the journal club right now. So the next parameter that I just want to mention is called R-free or free R. And through the history of the protein data bank, it has not always been an included metric for X-ray structures, but since 2010, so this plot shows bubbles based on the size of how many protein structures were deposited from X-ray, and what percentage of those had included this metric free R. So the free R is a measure of how well the protein structure was fitted to the electron density. So when you look at structures, you should check out the free R, that it's not too high. So a good rule of thumb is not to take the ones that are the worst structures and take the ones that are the good structures, which would be hard cutoff somewhere around here. So this is a hard cutoff that I use for the data that I'm working with. But you can see how the free R follows the resolution somewhat. So the better the resolution, usually the smaller the free R also is. And these graphs shows the interquartile range. So the lowest one is the top quarter of structures, and the yellow one is the worst quarter of structures. So what is currently being done with the protein structures? So I just want to highlight these two to show that there are actually some really good existing efforts out there already. So this one to the left, it's from Higher News Group in Cornell, and they use protein domains, and they look at different disease genes, omin, and I think they also use ClinVar. And the other one here, disease map, is from Patrick Aloy's group in Barcelona. And those are also some really interesting paper that I would recommend you to check out. But I would like to narrow in now on some of the things that are not so much available in the existing literature as far as I know. So one simple thing, going a little bit back to the start, when we have interactions between atoms, between proteins in particular, we have these 4 different types of pairs. So covalent bonds and salt bridges, hydrogen bonds, and non-bonded interactions. And these 4 different types have all been classified and are in this database called PDB-SUM, which makes a summary of each single PDB file. And to show you what these 4 different types looks like, I have just showed you a little cutout of a protein structure for each example. So here is a covalent bond. That really only happens when you have cysteine making a disulfide bond, and the 2 atoms are actually sharing electrons. And so it actually becomes one molecule. The next one is salt bridge. What you see here is that you have an electrically charged group on one side and an electrically charged group on the other side, and they will attract each other quite strongly. So what you see here is a lysine to aspartic acid. The hydrogen bond is also an attraction, is a little weaker, but basically you have a lone pair of electrons on one side, and you have protein on the other side, and they bond the same way that water bonds with each other. Then you have a non-bonded interaction where the van der Waals forces are in place because the proteins are so close to each other. So if we rank them by attraction, we can very simply say that the strongest ones are covalent bonds, salt-bridged, and hydrogen bond, and the non-bonded interaction. And if we plot this against the constraint scores, we'll see something like this. So the constraint scores, again, this was presented earlier in the journal club. So these are Missense constraints. So if you don't remember, it's a measure of how constrained, in this case, amino acid is to mutate based on the observed mutations in exact and predicted mutation rate. So what you see is that there are more constrained amino acids that happens in the stronger bonds than in the weaker bonds, which is interesting because we can just detect what kind of interaction happens from the protein structures. And the example shown in the beginning is actually a hydrogen bond type. So a very old feature in the field of protein structure is secondary structure. And I found this cover of a paper from 1983 from Chris Sanders. And this is a method to identify secondary structure and also surface contribution of a single amino acid. So the secondary structures, they would be, is the protein alpha helix, or is it a beta sheet, or is it a turn? And there is an updated version recently, which has 8 different types of secondary structures. So those are 3 different types of helixes. The very tight and the more loose ones are included. And these ones are quite useful because we can get a lot of these basic features. So I've been looking into a metric called frustration, and I'm sorry for introducing a lot of different concepts, but the frustration is actually quite interesting because the paper describes a way to make a lot of decoys, so a lot of alternative protein structures optimize their configuration, and for each amino acid figure out how much freedom does that amino acid have to move around. So amino acid in a protein that has freedom to move a lot around would have a low frustration, and an amino acid that is very, very contained in space would have a high frustration. So that the idea is that the amino acid would be frustrated if it couldn't move. So if we take the exposure versus the frustration, we can see that the more exposed an amino acid gets, the less frustrated the amino acid gets, which is very good to see because we expect that an amino acid that is in the core of a protein would have amino acids all the way around and would have less freedom to move. One on the surface would have more freedom to move around as it's only covered by some sides. So this is interesting, and it's interesting not to see there's a perfect correlation, but we have a close to 0 p-value in a t-test here. So this is a feature that's interesting and we can use. So there's yet another concept called deep mutational scanning, where you can take and mutate all amino acids one by one in a protein. And this is very computationally intensive. So before doing this, we selected a subset of genes. So we did this with the top 1,003 most constrained genes in the paper of Caitlin Samopa. And we subsetted them of the ones that have a protein structure. And we've got this kind of interesting figure with Christian Davidson, a master's student from the Technical University of Denmark, has generated. So what is plotted here is whether missense mutation has been seen in the exact data set or not. And on this axis, what you will see is the delta Gibbs energy, or Rosetta energy in this case, since it's calculated in Rosetta. So it's used, we use the monomer protocol of Rosetta, which will mutate a single chain. And we did this for the amino acids that are in the core of the protein, so that are 100% boric. They don't have any surface contribution to the protein. And so what is interesting here, and you'll see, is that the amino acids that have not been observed in the exact data set, so the amino acids that have, the mutations that have not been seen in the healthy populations, they will take energy changes that are much higher than the ones that have actually been seen. So this, the right plot has only 94 data points, and the one here on the left have more than 3,000 data points. So maybe this line will go up if we get more data points. But we did the same with synonymous mutation, and it was not significant, which was interesting to see. So yeah, this has kind of been a summary of some of the features that exist and can be extracted, and focusing on the ones that I have been working on. I just want to mention that I've also been extracting distances from each amino acids to sites with post-translational modifications, as well as some functional annotations from UniProt. So all of this is so far in an internal database, which I plan to release and publish, so people can take advantage of that at some point. So I want to say thank you to the lab, and thank you for Dennis to organizing. And if any of you have any questions, I'll be very happy to answer, or if you want to have a look at some of the data, I will let you know when the database is available, or I can run some of your mutations through the internal pipeline that I have available right now.